A very good afternoon to everyone and a warm welcome to the first of the Precision Nanosystem Tea Time webinar series for 2021. It's my pleasure this afternoon to introduce our speaker, Dr. Giulia Anderluzzi from San Raffaele University in Milan in Italy. And today's presentation will be very topical and I think very interesting for everyone, and it's entitled Rational Design of Cationic Nanosystems for the Delivery of mRNA-based and Subunit Vaccines. This is brought to you in conjunction with the Precision Nanosystems Europe team. So just to introduce us, my name is Richard. I work as field application scientist for the north of Europe, which means Scandinavia, Benelux, and the UK and Ireland. And working alongside me is our regional sales director and commercial leader for Europe, AJ Johnny. And also with us today for Central and South Europe, regional sales manager is Jürgen Schmidt-Sörktig, and my equivalent in the south and centre of Europe is our field application scientist Martin Rabel. And Martin will be conducting the question and answer session at the end of the seminar today. So I do hope you find today's presentation, I'm sure you'll find it very interesting. Please do interact and ask questions. It's now my pleasure to hand over to our speaker, Giulia Anderluzzi. Thank you.
So thanks, Richard, for the introduction. So hi, everyone. And today I would like to share with you my latest work on cationic nanosystems for the delivery of mRNA-based and subunit vaccines. And this was part of my PhD project. So the objective of this presentation is to screen among different cationic nanosystems to identify effective delivery system for self-amplifying mRNA and subunit vaccine platforms. So these lipid-based formulations were first characterized in terms of physicochemical properties alone or in combination with self-amplifying RNA or subunit or protein subunit. Then they were tested in vitro, where we tested the particles' transfection efficiency and promotion of antigen expression for self-amplifying RNA, and the association with cells and promotion of antigen processing for protein subunit. Then they were tested in vivo, where we evaluated both the pharmacokinetics and the immunogenicity of selected candidates upon intramuscular administration. So vaccine delivery using nanoparticles is a well-established way to increase vaccine performances. For example, they can be used as adjuvants to boost the efficacy of poorly immunogenic vaccines, or as a delivery system to protect degradable vaccine and increase transfection into the cells. In the literature, there is a variety of particles we can use. However, the most frequently described are essentially four. So polymeric nanoparticles, liposomes or LNPs, solid lipid nanoparticles, and emulsions. Of course, they are very different in terms of chemical composition, three-dimensional structure, rigidity, and all these elements inevitably affect vaccine efficacy. Therefore, to understand the impact of the delivery platform on antigen-specific immune responses, we formulated and compared these four nanosystems for the delivery of two different vaccine platforms: a self-amplifying RNA, abbreviated as SAM, and an Neisseria-Daisy A variant 3 subunit antigen, abbreviated as NAD3. So the first application of these nanosystems were to deliver a self-amplifying RNA. So briefly, SAM is based on an alpha virus genome having a positive single-stranded RNA, in which the genes encoding infectious viral proteins have been substituted with genes encoding the antigen of interest. However, the virus amplification machinery, for example, polyproteins involved in the replication of the RNA in multiple copies, have been maintained, therefore allowing some self-amplification and replication over time. In the specific study, we selected a SAM encoding for the rabies glycoprotein G antigen, abbreviated as RBG, as there are established correlate of protection for neutralizing antibodies, and there are available commercial vaccine against rabies we can use as comparators, such as Rabipur. So in contrast with DNA vaccine, SAM doesn't need to be delivered in the nucleus to be effective, as the antigen expression occurs within the cytosols, such that any potential genomic integration is avoided. Compared to non-replicative mRNA, SAM is more potent due to its ability to self-amplify and produce multiple copies of itself and of the antigen of interest. and it's able to induce both humoral and cellular immunity. However, as all the mRNAs is subjected to biological degradation, especially by RNases, and the delivery within the cytoplasm is quite challenging because due to its high molecular weight and high hydrophilicity. So there is a need for some to be co-formulated within a delivery platform to be applicable for clinical evaluations. So in this specific case, we decided to co-formulate some either inside or outside the particles to provide protection and increase cell transfection. So these are the 4 different types of particles we formulated, either using the nanoassembler platform or the microfluidization method. So polymeric nanoparticles based on PLGA, emulsions based on squalene, liposomes based on diffusogenic lipid DOPE, and solid lipid nanoparticles based on tristerin. Although they were very different in terms of chemical composition, they all contain the same amount of non-ionizable cationic lipid, either DOTAP or DDA, which increase some association, so some encapsulation and or absorption, and increase the association with the cell membrane and the consequent transfection. So first, these formulations were chemically characterized, physicochemically characterized. So this is a lot of data, and I'm not going to go much into details, but I just want to point out that with using the nanoassembled platform or the microfluidization techniques, we were able to formulate 4 different kinds of nanosystems with an average diameter between 40 and 150 nanometers, depending on the formulations, low polydispersity index, and cationic surface charge. The addition of some either inside or outside the particles increased particle size up to 200 nanometers, depending on the formulation again, without affecting the polydispersity and the surface charge. For all the formulation, we achieved an encapsulation efficiency or absorption efficiency above 90%. Then we proved that the formulation offered protection to some from RNases degradation. So as you can see here from this representative agar gel picture, we observed that when unformulated SAM is incubated with RNases, it gets easily degraded. However, when it's co-formulated within a delivery system, it gets protected. So this is just an example with DDA, liposomes, entrapped in SAM, but we observe a similar trend with the rest of the formulation as well. Then we move in vitro, where we evaluated the potency of these nanosystems to induce transfection into fibroblasts and produce antigen expression in the HK cells. So within the, so we incubate the particles for 16 hours and we use lipofectamine 2000 as positive control. So within the SAM concentration tested, we observed that all the formulations were actually able to transfect the cells and induce antigen expression. However, among the candidates we tested, we observed that DOTAP nanoparticles entrapped in SAM and both DOTAP and DDA liposomes entrapped in SAM were the most effective in inducing consistent antigen expression in the HK cells. So given these encouraging in vitro results, we decided to move this formulation forward into an immunogenicity study. So basically, we vaccinated mice intramuscularly twice, 4 weeks apart, with 2 SAM doses, so 1.5 and 0.5 microgram. And we compared the antibody response with a benchmark for SAM delivery, which is CNE56, which is an emulsion-based system now being tested in phase 1 clinical trial, and with a commercial vaccine for rabies, Rabipur. So on the left-hand side, you see the antibody response 4 weeks post-prime, and on the right-hand side, you see the antibody response 4 weeks post-boost in sera. So looking at the prime, we observed that all the candidates we tested were able to actually induce a measurable antibody response. However, polymeric nanoparticles were the most potent in elicit antibody response. And most importantly, the GMT, the geometric mean titers at both SAM doses was above the correlate of protection of 0.5 EU per ml, which is represented by this red line here. So 0.5 EU per ml is considered a positive protective antibody response to rabies virus according to the WHO. Therefore, the titers above 0.5 EU per ml can be correlated with functional neutralizing antibodies. Moreover, the potency of nanoparticles was comparable with that of the CNE56 at low dose and the vaccine on the market. After the boost, we observed a similar trend. So although all the formulation increased in titers, polymeric nanoparticles were the most potent and at both some doses. And again, the GMT was protective and the potency was comparable with that of CNE56 at low dose. Similarly, polymeric nanoparticles were the most effective at inducing proliferation of splenic polyfunctional CD8 positive T cells, especially at high SAM dose, and the frequency was comparable with that of CNE56 and with the vaccine on the market. So the second application of these nanoparticles was to deliver an Neisseria-daisy A variant 3 subunit antigen, abbreviated as NAD3. So briefly, NAD is a homotrimeric adhesion protein involved in the host epithelial cells adhesion and can mediate colonization and invasion. It's able to induce titers of bactericidal antibodies and is now one of the components of the Bexsero vaccine, which is a licensed vaccine against MenB. So the reason why we choose NAD3 as model antigen is because the immunogenicity of the protein is significantly boosted when co-formulated with an adjuvant, and there are available data from the crystal structure and mapping of the binding site of human bactericidal antibodies. So in this case, we only selected DDA-based formulations, as is known by the literature that DDA is more immunogenic than DOTA, and we only absorb the antigen on the particle surface, as the conjugation of the antigen on the particle surface can allow the presentation of the antigen to the immune system in a similar way that it would be presented by the pathogen, thereby provoking a similar response. So again, by applying either the nanoassembly platform or the microfluidization method, we were able to formulate 4 different cationic delivery systems with an average diameter around 100 nanometers, low polydispersity, and cationic surface charge. Once again, the addition of NAD onto the particle surface increased particle size up to 350 nanometers, depending on the formulations, without, however, affecting the polydispersity and the surface charge of the particles. So in terms of encapsulation efficiency, we obtain an encapsulation efficiency above 85% for all the candidates tested. Then we move in vitro, where we evaluated the ability of these particles to associate with THP1 macrophages and promote antigen processing. So to measure the association, we fluorescently labeled the formulation with the DID probe. While to measure antigen processing, we substitute NAD3 with DQ-OVA, which fluoresces upon hydrolysis of OVA. So if we look at the cell association expressed in terms of mean fluorescence intensity, we observe that particulates, so liposomes, polymeric nanoparticles, and solid lipid nanoparticles readily associated with THP1 macrophages after 4 hours incubation, while emulsions were less effective. And we also observed that there was no significant difference in the way the particulates associate with cells. However, if we look at the actual number of particles which associate with cells, we did observe a difference. More specifically, liposomes tended to associate in a greater extent compared to both solid lipid nanoparticles and polymeric nanoparticles. In terms of antigen processing, we observe a similar trend of what we observe for cell association. So once again, particulates induce higher antigen processing in vitro. However, the efficiency of emulsions to induce antigen processing was comparable with that of unformulated DQ-OVA. In vivo, again, emulsions behave differently than the rest of the formulations, and in particularly in a pharmacokinetic study, while liposomes, solid lipid nanoparticles, and nanoparticles form a depot at the injection site upon intramuscular administration, of both the adjuvants and the antigen, emulsions were drained much more rapidly from the injection site and did not provide a longer permanency of the antigen at the site of injection. And this is confirmed by the image of the mice reported on the right-hand side. Then we wonder whether we could have a different accumulation at the lymphatics. So to perform this biodistribution study, we radio-labeled the formulation using a tritium cholesterol probe. And in this case, we observed that emulsions accumulate much faster and in a greater extent compared to the rest of the formulation at the popliteal lymph nodes. For example, if we look at the 24 hours post-injection, we see that the percentage of emulsions is at almost 3 times higher than that of the rest of the formulations. While we did not observe any difference in terms of percentage at the inguinal lymph node, of the particles at the inguinal lymph node. Then we tested this formulation into an immunogenicity study. So again, we vaccinated mice intramuscularly twice, 4 weeks apart, and we collect spleen and sera 2 weeks after boost. We use only one antigen dose of 10 microgram, and we use unadjuvanted NAD3 as positive control. So we'll look at the total IgG titers first. So as you can see here, conversely to what we observe in vitro, in vivo, emulsions behave very well. And in particular, they were the most potent formulation in inducing total IgG titers compared to the rest of the candidates, while polymeric nanoparticles were the less effective. And the potency of polymeric nanoparticles in inducing IgG titers was as good as unformulated NAD3. Then we look at the IgG subclasses, in particular IgG1 and IgG2A. So if we look at the IgG1, we observe a similar trend of what we observed for total IgG. So once again, emulsions were the most potent in eliciting IgG1 subclass, while polymeric nanoparticles were the less effective. Overall, we measured higher titers of IgG1 than IgG2A, and we couldn't measure a detectable concentration of interferon gamma response. So altogether, this data means that the antibody response induced are strongly polarized toward a Th2 type response. Sorry, it's not going, it's not working. Oh, okay. Yes, this was the interferon gamma response we couldn't detect in spleen. So in conclusion, we can say the application of microfluidics or microfluidization resulted in narrow size and monodispersed cationic particles with highly positive electronic dense core. And all these attributes are suitable for preclinical and clinical applications. In vitro, some polymeric nanoparticles and liposomes induce the highest antigen expression, and among them, polymeric nanoparticles were the most effective delivery system for some. As for NAD3, in vitro polymeric nanoparticles, liposomes, and solid lipid nanoparticles show the highest cell association and antigen processing. However, in vivo, emulsions were the most potent adjuvant for NAD3 among the candidates we tested. So this was part of my PhD project made in collaboration between the University of Strathclyde and GSK Vaccine in Italy. So I just want to acknowledge my supervisors, Professor Yvonne Perry at University of Strathclyde and Dr. Barbara Bautner at GSK, all the team at University of Strathclyde, GSK DP Discovery Team, GSK Advisors, GSK Preclinical, and all of you for the attention.
Thanks a lot. Thanks for your presentation. Thanks a lot for answering all these questions. And I would like to thank all of the attendees for coming to our Tea Time webinar. Stay tuned. We have the next one coming up in 3 weeks. And yeah, I wish everyone a great week and see you next time. Goodbye.
Bye.